Okay, now, today is fetal pig urogenital system. That covers the excretory system, which, while there is some component in the skin for that, what we're talking about really is the kidneys and the ureters and the bladder. And after that, it's the female and male reproductive systems, which is going to be one large exercise in trying to find which of the small tubes that look the same is which. Hopefully we haven't eviscerated our pigs too much so we can actually find some of these. But your best reference is looking at the pictures in this PowerPoint carefully and the very well dissected pigs behind the screen at present. Make sure you get a look at both of those. So, like I said, in terms of the large-scale things you need to know about what the excretory system, the urinary system is, there's really only the three. So, kidneys up high. We also need to know some substructures that go with that. You need to be able to identify the ureters, which plug into the urinary bladder, and the best thing to identify those is actually where they join up with it, which is kind of the more superior position once we get into our pigs. Notice that a lot of this stuff is buried in the pelvic girdle. So to actually get at the pig, we're going to have to get in there and pull it out. Okay. So when we made our incision on our pig, we made this kind of U-shaped cut around the urinary bladder and the umbilical vein and arteries, and so it's this nice muscular thing right in the middle of it. You'll notice the ureters coming in right at the top of it. So again, one of the things we'll do today is try to find a good structure to start with, and then we'll trace back from it. So the ureters are the tube that carry the urine away from the kidneys, and you can find them meeting up here in the hilus and coming down and meeting up right here. Again, there's a lot of tubes that look the same, so you need to be able to distinguish which one's which. Things you need to know about the kidney are kind of the macroscopic components to it. So you need to know the vasculature, you need to know vein and artery, you need to know the biggest duct moving away from it, the ureter, which carries the urine. I don't think that these guys here are important, but you should know the pelvis or the hilus is where it all comes together. You should know that the pyramids are these kind of smaller subdomains in there. And most importantly, bright flashing lights, know that the cortex is the area on the outside and the medulla is the inside. And that's where most of the dialysis occurs. So the major function of the kidney is homeostatic. It's to take improper amounts of salts and water out of your system and get your blood back to where it needs to be in terms of the concentration of those things. Okay? The structure is heavily vascularized. So actually we don't have a slide in here, but you should find a figure, internet, or I have an anatomy book here with me in the lab. You should also know this. The functional unit of the kidney is called a glomerulus. Excuse me. Sorry, that's one part of the functional unit. Scratch that. The functional unit is called a nephron. And what that is a collection of tubes with direct access to capillaries, kind of like an alveolus in the lung. It's kind of a similar arrangement. But then hooked up to a bunch of pipes that look like the drainage pipe on your toilet, that kind of loop. And it kind of looks that way because it's kind of got the same function. It's to wick away what we decided we did not want. And if you want to be fascinated by how your body structured something, look up this concept of countercurrent exchange, how your body is very efficient at getting the maximum amount of water or salt out of a solution and concentrating it for secretion, or excretion rather. Okay, so we want to be able to follow the path of our gametes from where they're produced to how they get to the outside, or what's functionally the outside, I suppose, for well, yeah, let's change that. To where they get to where the two gametes can meet, right? On a woman, obviously, the gamete's going to stay on the inside. So those relevant structures are the ovary, where we've already looked at in tissue section, where the eggs are produced. There's actually kind of an indirect way into the oviduct. You need to identify that as it becomes the uterine horns, which is a term we'll use in this class, which is very confusing when you go across species. Before I get to that, 1 second. Uterine horns come into where they meet up. That's the uterus proper, and what we'll compare to humans by that name, which is then bounded by the cervix. There's a muscle that prevents anything coming out of the actual place where the fetus gestates, and the vagina where we have access to the cavity. And then all the fun parts on the outside aren't necessarily important for our class because we're talking about the reproductive aspects. So one of the major things, there's some slides of the fetal pig following this. We'll try to point out the correct tubes going in the right place. Again, look at the dissected pig. But one of the really confusing things is different mammals, different vertebrates that have this internal gestation use different parts of these tracts for the actual place where you're going to implant the embryos. A mouse or a pig will let the fetuses attach to the actual uterine horns. Some is in the oviduct, and in humans it's in the uterus proper, because we're in the habit of only having one kid at a time. So if you look at the difference in the anatomy of these structures between those different species, it reflects where we're going to let the kids grow up. So in a pig, you're going to have a big uterine horn because you're going to have rows of fetuses growing on each side. If you're a human, you're going to shrink those mightily. You're going to have a big uterus where you're going to host one fetus. Okay? You should be able to look at the human system in cross-section in cartoon, and then there's models over here, and make sure you don't get it incorrect what's actually the colon versus some of the more relevant cavities for reproduction. So in the fetal pig, you should be able to identify the ovaries as these lumps here. Connected to them, you should be able to see the beginnings of the uterine horn. We're not going to distinguish the oviducts from those, but then you'll see them meet up at the center. That's what we'll call the uterus. That's actually dissected very well on the pig in lab. And then the only way to identify the other structures is just distance from the uterus toward the actual urogenital opening. And so you would then, if you can find the muscle, that's the cervix, but then the vagina is behind that. Try not to confuse these. The umbilical arteries are very prominent, but say the ureters might look almost exactly this way, so you really have to pay attention to how they're connected to make sure that you get these two structures correct. Okay. The ureters should be plugging in to the bladder. This is the tail end down here. They should be plugging in right about here. So you see that they're almost in about the same spot. Yes, and here's the detail on those subuterine horn structures. Okay. The male, we have the complication that not all the things that we're interested in join in a linear fashion. I will explain. So we have production of the sperm. Again, we've covered that histologically. That happens in the testis. They mature in the epididymis. Yes, you do have to remember all the stuff we told you last time about all the hormones that come from these places, what cells produce them, and so forth. So, yes, we do retest you on previous material. No, you are not allowed to answer BS when those questions come up on the tests. Fair deal. So that we've all had before. Notice that the wiring is funny. The vas deferens, which carries the sperm, ultimately to the final destination where we'll get to the ureter and excreted, loops up around where it joins with the first of the glands that we're going to concern ourselves with. That's the seminal vesicles. Let's see, seminal vesicles are bilateral. There's 2 of them. I don't think that we'll observe them in the pig. They're responsible for the bulk of what we would call semen, the fluid that is. It's just general things to make sperm happy, including a little alkalinity, so they make it more basic, so that they can better survive their travels through the more acidic vagina and uterus. Okay. After that, we're going to add some zinc and other stuff with the prostate gland. That comes after. Finally, the bulbourethral gland is closest to the actual linking up with the urethra down here, and that's essentially a way to lubricate the actual shaft of the penis so that the sperm can get out. Okay. So those 3 glands in that order are important for you tracing on a test the production of sperm to its release. We go from the testis through the epididymis, through the vas deferens, meeting the seminal vesicles and the fluid that comes from that, then meeting the prostate gland, then a meeting with the cowpers, or the, I'm sorry, what's the term that we use again? Say it again. Bulbourethral glands. Thank you. Different names throw me. Out the urethra and out through the penis. Again, be able to identify these things in cross section and on the pig. Let's take a look at the wiring. You can see that this is a bunch of very fine tubes, and again, the best way to identify them is to see what's connected to what. You can actually dissect out your testis so that you'll get a distinct lobe for the testis itself, a nice loop for the epididymis where they mature, and from that we have to find something that connects well enough to actually identify the vas deferens, which they don't even show here. Okay, they just show it coming out along here. There's of course vasculature that'll get in the way too. Again, there's a challenge in not confusing it with the ureters, which again meet here at the more superior position on the bladder. And I think the next one gives us a chance to see some of the other. Yeah, so the bulbourethral Cowper's glands are down here. They're closest to the actual point where we meet with the penis. You might have to work backward from the exterior to actually find the penis versus, and to distinguish it from the bladder and such. And again, the other structures, they're probably too minor to properly dissect for our purpose in this animal. But that's the tour.